So good evening, everybody. Good morning, everybody. And good afternoon for those, and good middle of the night for those in Europe. Believe it or not, there were some people from Europe watching last night. It's a great pleasure to open the second day after a very successful day yesterday of this third Global Microbiome Network, GloMiNe, Symposium this year for Asia. And we have three great speakers this morning. The first one is Jack Gilbert from the Scripps Institute of Oceanography, University of California in San Diego. The second one is Yusheng Zhen, from Westlake University in Zhangzhou in China. And the third one is Jiro Nakayama from Kyushu University in Japan. So, and this session is the human microbiome. So without further ado, it's my big pleasure to introduce my friend and inspiration, Jack Gilbert. Jack, the podium is yours.
It's a great pleasure to be here. Thank you very much, Maria Gloria, for that wonderful introduction, and I'm really happy to be able to speak to a diverse audience across the world. I think this is a great example of why Zoom is still a powerful tool. So I'm at University of California, San Diego, as Maria Gloria pointed out. I'm actually in the Department of Pediatrics and the Institute of Scripps Institution of Oceanography. I'm probably one of the world's first pediatric oceanographers. I'm also the PI and director of the Microbiome and Metagenomics Center at UCSD and faculty member in the Center for Microbiome Innovation. So I also just wanted to highlight, I'm the incoming president of the Applied Microbiology International Society. And we are a society, one of the oldest microbiome societies or microbiology societies in the world. Our primary drive is to enable members to access ways of manipulating government policy in different parts of the world. We have active grants and funding available for members, professional development and educational development available for members. And we are aimed at helping to solve the world's biggest challenges, produce networking and events, and we have a series of journals which we publish. So if you're interested in getting involved in societies, which I strongly suggest you do, then please find out more at appliedmicrobiology.org. So I want to start out my lecture talking about human microbiology, but I talk about human microbial ecology. I trained as an ecologist, and I'm very engaged in trying to understand how the human microbial ecology interaction shapes human health. And I like to think of this in two situations. Number one, the microbiome is there at the behest of the immune system. The immune system evolved in order to handle microbial exposure. It's impossible to avoid microbial exposure because we, as a eukaryotic multicellular life form, live in a world of microbes. It's a microbial planet. And so we had to evolve an immune system to act like a national park warden or a gardener that can manipulate the growth of certain organisms, remove weeds, bad plants like bacteria, and promote the growth of bacteria, which will provide us services, much the same way as a farmer or a gardener would manage their farm or their garden. And so this is really important. When this relationship between the microbiome and the immune system breaks down, that's when we have illness and disease. A great example of this was provided by Mariana Bindlos, who's a fantastic now associate professor in the United States. And she provided this infographic in a paper from Microbial Ecology in 2018, where she highlights the differentiation at a mechanistic level between homeostasis and dysbiosis. So if we consider these as cycles, positive and negative feedback cycles, we can really understand what we mean by these two terms. Homeostasis is where here I have a low oxygen potential in the gastrointestinal cavity. I have, therefore, a promotion of the growth of obligate anaerobic bacteria, which ferment dietary fiber to produce short-chain fatty acids, SFAs here. Those short-chain fatty acids, like butyrate, are a food source for developing colonocytes that line the gut, powering a beta-oxidation cycle, which consumes oxygen out of the bloodstream to maintain a low oxygen potential in the gut, and also to drive T regulatory cell accumulation and therefore a moderation of inflammation. This is a positive feedback loop because that oxygen consumption keeps the oxygen potential in the gut low, which allows these obligate anaerobes to produce the short-chain fatty acids to allow the colonocytes to mature. And this cycle keeps on going. It's a bidirectional homeostasis cycle. If we gave our patient an antibiotic or fed them a very high-fat, high-sugar diet for long enough, we would get what we refer to as dysbiosis, a removal of these obligate anaerobes and a promotion of the growth of facultative anaerobic bacteria that can survive in the presence of a higher oxygen potential. These bugs do not tend to produce large quantities of short-chain fatty acids. They do not tend to ferment fiber. And so we actually see a reduction in the maturity of our colonocytes lining the gut and a promotion of anaerobic glycolysis by these colonocytes, which allows oxygen from the blood to seep into the body, into the gastrointestinal cavity, promoting the growth of facultative anaerobic bacteria that can survive without oxygen. These bugs also tend to produce lipopolysaccharides, LPS, and hydrogen sulfide and flagellin, which can be potent stimulators of inflammation. This is a negative feedback cycle, what we refer to as dysbiosis. And dysbiosis can influence many aspects of human health. This ecological disruption to the microbial community living in the body can influence things like obesity. So, for example, in this study we published now 8 years ago, time flies, we gave mice either a low-fat, high-carb, high-fiber diet, or a high-fat diet and high-sugar diet, and observed what this did to the oscillation in microbial ecology inside the gut over a 24 to 48-hour cycle. What we found is that in conventionally raised animals that are not germ-free, those animals that are fed a high-fat diet will have aberrant microbial oscillations. Their ecology will be quite disturbed compared to what we would normally see in a conventionally raised animal receiving high-fiber chow. And those aberrant signals are associated with the production of things like hydrogen sulfide and lipopolysaccharide, which increases in the bloodstream and disrupts circadian cycles in our body's organs, such as the liver, kidneys, pancreas, etc. And that disruption in circadian cycles, how our body's organs sense time, disrupts that relationship between the brain and the body, and actually can lead to an increase in fat deposition from food calorie intake, leading to an obese animal. So these high saturated fat diets increase the abundance of facultative anaerobes, as we saw previously here under a dysbiotic system, increasing hydrogen sulfide production, changing the hepatic hormones in the liver, increasing adipose tissue production and appetite in the animal. We've also demonstrated this from an ecological perspective, working with people who are of traditional African origin. In this case, we selected women of African origin in both the United States, represented by the U here, and Ghana, so represented by the G. And what I have here are 4 body types. So Ghanaian women who are lean, Ghanaian women who are obese, United States women who are lean, and United States women who are obese. And what we did is we took data from these participants in our study, and we networked the correlation networks of the microbial communities present in their guts, and asked ourselves a question: is the connectivity between the organisms in the gut of these people different? And indeed, lean Ghanaians had significantly greater network density, connectivity, and stability than either the obese Ghanaians, the lean, or obese US participants. This is represented by the statistical significance of this star here. This phenotype can be transplanted into mice using lean Ghanaian stool, and it was associated with weight loss, representing a significant increase in the network integration of the ecology of that microbial community. We hypothesize, therefore, that greater network connectivity and closeness between those organisms in their representation across people represents greater cross-feeding and ecological stability in the gut, suggesting that a high-fiber diet creates that ecological niche structure to provide a robust microbiome with dense metabolic interactions that can promote a healthy outcome in these participants. It's quite telling that lean Americans have a very similar level of network stability as obese American women. And we hypothesize this is because even lean American women have a disrupted dysbiotic microbiome that, in the presence of a high sugar, high fat diet, would rapidly shift ecological states, whereas a Ghanaian lean microbiome tends to be a very stable ecosystem that would be very hard to push out of that healthy ecological state because it is so robust. Working with one of my very talented postdocs, Gertrude Eklumansah, we went back into participants in this cohort across Ghana, Jamaica, the Seychelles, South Africa, and the United States, and examined the species richness of more than 2,500 people in these populations, and identified the lowest species diversity coming from countries like America and the Seychelles, and then subsequent to that, Jamaica and South Africa, which represent a gradient of social development, an acculturation gradient, if you will, in their developmental processes. The Ghanaian population tends to be much more rural, whereas the American and Seychelles population tend to be a much more urban environment. And we do see that Ghana has the greatest microbial diversity, as I pointed out here, likely resulting from a high fiber content in their diet. And when we actually look at which microbes are promoted in their proportion in the Ghanaian microbiome, we find things like Prevotella and Lactospira, which do ferment fiber and produce short-chain fatty acids. And interestingly, a significant increased concentration of Weissella, which is a lactic acid fermenter enriched in traditional African fermented foods found in the Ghanaian diet, suggesting that the foods they're eating, fermented and high fiber, are leaving a fingerprint in the gastrointestinal microbial ecosystem. People living in the United States, the Seychelles, South Africa, and Jamaica tended to be enriched in other organisms, which are much more commonly found in people living in an urban developed population. When we actually look at these populations, the microbial community in our Ghanaian population tends to be significantly associated with low blood glucose, low blood pressure, and a normal BMI. And when we look across the entire population, the entire global population, an elevated level of microbial alpha diversity, this term we're using Shannon index to compute the diversity of the microbial communities. No matter what country you come from, a higher level of diversity tends to associate with these markers of health: low blood glucose, low blood pressure, normal BMI. So while the differences have varied, overall diversity and ecological connectivity provides a correlation with lower level of diabetes and cardiovascular disease risk. Interestingly, we're also able to drive these differences in dynamic ways. And so, for example, here we can look at the microbial ecology prior to surgical intervention, relate it to human host behavioral factors like where you live, if you have furry pets, what you eat, and determine that that actually impacts the kind of infection state you're more likely to exhibit in this population. We found that people, a Western diet and lifestyle, promotes the colonization of the gut by collagenolithic microbes that promote tumor formation following colorectal surgery. So, for example, in this video, if I excise a piece of the gastrointestinal tract and remove it, I actually expose that gut microbiome to a lot of oxygen, which obviously knocks out all the obligate anaerobic bacteria and allows the proliferation of the facultative anaerobes. I then pump the body full of antibiotics, selecting for those bugs which have antibiotic resistance. And then when I stitch the gut back up and form an anastomosis, the body starts to leach phosphorus out of the gut, starving the bacteria, triggering a phenotype change in those collagenolithic organisms which are present, allowing them to bloom, form a biofilm, grow on the wound site, release their collagenase, break down the collagen, and lead to the opening of the wound site and the expelling of your gastrointestinal contents into your body cavity, leading to sepsis and death. So what have we been doing? Well, we've been exploring the microbiome from an ecological perspective just by looking at the phenotype of stool for many, many years. Now we're adding bacterial DNA, microbial DNA, to understand that phenomenon more effectively through what we refer to as microbiome-wide association studies. But however, we're at a crossroads. Our traditional methods using cross-sectional analysis at a massive scale through something like the American Gut Project, which Maria Gloria has been involved in, identifies associations which can link microbial metabolism to how we process the primary metabolites, drugs, antibiotics, and body chemistry. Great. But we're also missing a component in the way of time. So, for example, this study demonstrates that the more consecutive stool samples you microbially analyze, the higher the likelihood is that you'll find a significant biomarker in your population with the more samples you collect. Why is this so important? 1 minute, yeah, I'm finishing. Why is this so important? So things like height, weight, and age are static and low dimensional. Things like insulin and blood pressure are low dimensional, but they're very dynamic. They change hour to hour. Your human genetics is static but high dimensional. The microbiome is the worst case scenario. It's high dimensional data and it's highly dynamic. So you need sufficient cohort sizes to account for high dimensionality and frequent sampling to account for the changing ecosystem. We have built a new device that we've licensed out into a company called BiomeSense that allows for continual collection of the microbiome in the patient's bathroom up to 90 consecutive days, running off a battery, extracting all the DNA and RNA and metabolites and locking it in and preserving them for future analysis. We're deploying this in our Microbiome Metagenomics Center through the NIH's $170 million Precision for Nutrition study to launch this in 200 individuals undergoing a clinical dietary nutritional trial. So the microbiome can do a lot for us. I'll stop there. And yeah, thank you very much.
Thank you so much, Jack. Great talk. I learned a lot of what you're doing lately, and that device sounds incredible. We'll have questions for you there.